Thank you, Fahim. Hello again, everybody. I'm Sharmili, currently working on the project development of a biosensor for measuring odorants in the ambient air near solid waste management facilities. As of now, there is no analytical technique that can measure the intensity of the odor objectively at landfill at low cost. So it is very difficult to detect and quantify odor at landfills and industrial facilities. Our research focuses on detecting the odorants that are typically encountered at landfills and finding out their quantitation range by using human odorant binding protein as biosensor. Although we would be starting out with testing it for hydrogen sulfide, we would extend it to other model compounds like ammonia and finally to a mixture of gases that are actually found in landfill. I think this project has the potential to make odor investigation more manageable for landfill operators. There is a need for methods to measure the odor objectively at landfills, and we believe that we have the potential to develop that novel technology. Our research objective is to modify the human odorant binding protein into a biosensor by tagging it with a fluorescent marker or fluorophore. And then we will expose the biosensor to some selected model odorants to determine a positive response and concentration dependent by means of spectrophlorimetry. If human odorant binding protein can bind with odorants in the micromolar range, there is a chance that the protein will show an objective response if we mark it with a fluorescent marker. Since our neuron system processes the odorant intensity based on the number of odorants, so there is a chance that the protein response will be concentration dependent. We are expecting something like more the fluorescence intensity, less will be the amount of odorants. And there is another thing is that we can regenerate our biosensor complex by changing slight amount of pH. I mean, if we change the pH of the protein sample, the protein will release the fluorescent marker and will return back to its previous pH to do another type of measurement. Human odorant binding protein is found in the nasal mucosa of all human. It communicates with the brain by collecting the odorant to the neuron, olfactory neuron, through mucus. It is stable to temperature, organic solvents, and proteolytic digestion, and it has broad binding affinity. And it can also be expressed in bacterial system at low cost and can be easily purified. I think some of you are already familiar with the mechanism of how human odorant binding protein will work because of our previous project regarding hydrogen sulfide. I'm just going to give you just a short review of it. As you see in the slide, there are mainly three components of our biosensor. One is the odorant binding protein, and the other one is the fluorescent marker or the fluorophore, and the last one is the odorant. When the human odorant binding protein is attached with the fluorescent marker, and if we shine a light on this, the object looks like it's glowing from within. And we can measure the intensity of the fluorescent by using a spectrophlorimeter. And when this object is exposed to odorant, the protein will release some of the fluorescent marker and instead bind with the odorant. As a result, the fluorescent intensity decreases. So you see there is an inverse relationship between the fluorescence intensity and the amount of odorant present in the protein sample. Dr. Bininger, a professor from biology at Florida Atlantic University, has already supervised the production of the first large batch of the purified protein. His research team is already in the process of verifying the purity of the protein, and they have definitely made it easier for us to work with the protein sample. As soon as the protein is ready, I will convert them to biosensor complex by tagging it with a fluorescent marker. And the fluorescent we will be used here is the 1-aminanthracene, which was previously used successfully with the experiment for hydrogen sulfide. And finally, we will add a buffer solution to this mixture. The left one, the left picture in the slide that you see is the first prototype of the gas reactor chamber that was used previously. The aqueous protein sample was at the bottom of the bottle that you see in the middle, and there is a gas cylinder which is used to deliver hydrogen sulfide to the gas reactor chamber by means of a finely adjustable one-way check valve. You know, there are several means to develop this gas reactor chamber. I mean, we would definitely try to develop it for better experimentation. The sample bottle you see in the middle, we need to open it up and insert a pipette to collect sample in a cuvette for spectrophlorimetry. So you see, there is another sample bottle right over there, right up there, which has a spigot towards its bottom. If we use that type of sample bottle, we won't need to open up the bottle to collect sample, and we can deliver directly the sample to the cuvette. That's how we can reduce the possible contamination, if there is any, due to its exposure to the external environment. And for experimenting in a broad range, I mean, when we will work with the real landfill sample, we can use this type of cuvette right down there in the slide. This cuvette has a tube connected with it that can deliver sample directly to the gas exposure chamber by means of a peristaltic pump. And it will absolutely give us no chance of contamination due to its exposure to an external environment. We will begin our experiment with hydrogen sulfide, though it was tested before. We will just redo the exact same procedure for the verification of the result. We will expose the biosensor complex to hydrogen sulfide at incrementally increasing concentration. And at each concentration, we will measure the fluorescence intensity by means of a spectrophlorimeter. And then we will develop a linear model and verify the result with that of the previous one. And if we can establish a linear model for hydrogen sulfide with certainty, we can be able to measure any unknown concentration of hydrogen sulfide by just getting the fluorescence intensity. Let me show you a previous result for hydrogen sulfide. Here you see from the graph that there is an inverse relationship between the fluorescence intensity and the amount of hydrogen sulfide present in the sample. The black line you see there, the fluorescence intensity decreases in a linear fashion with increasing amount of hydrogen sulfide, but it began to deviate from this trend from above 5 microgram of hydrogen sulfide, which is showed by the red mark. There might be several possible phenomena for this deviation. One possibility is that the biosensor complex was becoming saturated at that point and reaching at a point of equilibrium, and it causes the fluorophore to release the protein. And other reasons would be relative binding affinity of competitive gases, or it may be just a sampling error. And we need to verify the reasons. Our previous result showed that the optimal ratio for protein and the fluorescent tag was 1:1. We need to verify that ratio. Also, we need to verify the accuracy of the calibration and the quantitation range by testing with different mixture of protein and fluorophore, by exposing the system to higher or lower concentration of odorants. We believe that more data and more trials would definitely strengthen the predictive power of the model, and we would also verify what will be the lowest concentration of hydrogen sulfide that the system can detect. After experimenting with hydrogen sulfide, we will check if the protein shows a binding affinity only towards hydrogen sulfide, or it will apply same with the other model odorants like ammonia. And if we find that positive, from that on, we will try it with a mixture of these gases. And we would also check how the acidic and the basic odorants would affect the binding of the biosensor complex. I mean, how the pH change would affect the binding. We would do more experiment for the verification of the results with hydrogen sulfide, and in future we will test it the same procedure with ammonia and other model compounds, and finally with a mixture of these gases. We would obviously try to develop the previous old conventional methods. So if you have any suggestion or advices regarding to it, please let us know. We would definitely try to follow that. Thank you. If you have any question, you can ask.
I have a question. Last one. I think your work is very interesting. You measured a protein, a specific protein, human binding protein.
Yeah.
Is that specific to hydrogen sulfide, or does it measure a lot of different chemicals, or is it only for that one chemical? I was just curious as to how specific that was.
Actually, we need to verify that. We already have that research with only hydrogen sulfide, and we will expose the biosensor complex with other pure odorants like ammonia that are commonly found in landfills. We need to verify that. Okay.
Oh, okay. Sorry about that. So it sounds like you're focusing on the gases that have odors, like hydrogen sulfide and ammonia. What about gases that don't, like methane? Does this protein work on something like that? Because that would be very useful.
We would also check that because the landfill contains a mixture of both odor and odorless compounds like ammonia, hydrogen sulfide, carbon dioxide, nitrogen, methane too. So we will check that.
Yeah, I don't know if you know about the air regulations out there, but we have to monitor landfills. They call it sweeping, and we use a meter, and we have to be below a certain threshold for methanes. It's 500 ppm VD. So another way of detecting these gases would be very interesting. I mean, right now we're just using meters, but we have regulations to meet. So I don't know if you guys are aware of that.
We would definitely try to.
Okay, thank you.
Thank you. Any other questions?
Okay, I think the only thing separating us from lunch is Bisho, so we will go to the next presentation.
Thank you.
Thank you.
Hello everyone. Am I too loud? Okay. So I'll talk about the investigation of leachate management solution at the Solid Waste Authority of Palm Beach County. Although the name looks fancy, but I'm going to talk about leachate logging. So leachate logging is a phenomenon. It has been all over USA from early 1990s, and there has been a lot of research about it, how it's forming, how to control it. But anyway, there were major three reasons of forming this leachate logging. As we found out in the research, the one is chemical precipitation, and one is chemical precipitation induced by microbes, and the third one is a combination of both. So these are the three reasons every researcher until now addressed the reason for chemical precipitation or biological leachate logging. Now, we have been studying this in Solid Waste Authority from several years, and we're also looking into this, but we wanted to know how this is exactly forming, and we wanted to give some insight and also how to manage this, give some idea to the Solid Waste Authority, as well as learn ourselves how to manage this. So my research focuses on how to manage leachate logging, as well as to understand the basic parameters or factors that affects leachate logging. So I'll show you today, I will walk you through my research process, and it is divided in 4 or 5 tasks. And I will go through the initial results, what we have, and what we're planning to do. So this has been developed between 2013 and 2016. One of the study we have conducted in Solid Waste Authority, that we have conducted water quality analysis for different leachate, and we calculated the Langmuir saturation indices, which is an indicator for the leachate precipitation potential for calcium carbonate. And yeah, I think it's needed to mention that the leachate clogging, it's mostly calcium carbonate. It has very high concentration of calcium as well as carbonate in the leachate, which forms calcium carbonate precipitation. It's pretty easy for calcium carbonate to precipitate in the leachate. So one of the way of measuring precipitation potential is calculating Langmuir saturation indices, and it's pretty useful for researchers as well as all the operators. So we calculated these Langmuir saturation indices for different leachate collected from different location, as well as some other waste flow that comes into the leachate mix. And we found out that very interesting is that NEFCO water that coming into the injection wall or leachate collection storage system before just a drip injection, it has very negative. It has negative LSI. So the negative LSI means it has corrosive properties on calcium carbonate precipitation. The positive, it's between the LSI fat is between, if it is 0, it's the neutral. It's neutral, which means it doesn't have any precipitation potential. Whereas if you go negative, it has corrosive properties. If you go positive, it has clogging potential. Now, it has negative, when NEFCO water has a negative precipitation potential, it has potential to be used as a dilution water or replacing the groundwater we use as a dilution. It has this potential. So we found that interesting and go ahead in the lab, and we try to do some mass balance analysis on that in the lab and see how that affects or how that improves the water quality to prevent leachate clogging. So we did some mass balance on based on the leachate volumes we see in monthly leachate volume we see from SWA, and we varied the leachate volume between 20 to 90 GPM, where we also varied the NEFCO water volume between 50 to 125 gallons per minute. And as we have seen in the wastewater NEFCO water quality, the pH varied between around 5. The pH is around 5 of NEFCO water. So we ran this analysis for 3 different pH at 4.5, 5, and 5.5. Now, if we see the results here, we see here that at 1-to-1 ratio of leachate and NEFCO water, it at pH 5, the LSI is about 0.5. We consider that 0 to 0.4 is neutral LSI, and it has very good potential of using as a alternative of dilution water rather than groundwater, which helps us as based on economically, as well as we can use this groundwater in other purposes from a point of operator's view. Now, the only problem with this NEFCO water is it has very high solid content, and these are sticky solids, and this leachate already has a precipitation potential. Now, if we add more solids in the system, it's become difficult to handle, and it can causes more leachate clogging, and it can be a disaster. So we don't want to really give an idea to a solid waste operator that could be a disaster. So we wanted to check that in the lab, and we tried to find out some ways to separate the solids from this NEFCO water before we add that as a dilution, as in dilution purpose. Now, there are different ways, and one of the ways cyclone separator, but we wanted to purchase one of the unit, but the units available commercially is too big for us to run in the lab. So we wanted to build one, a small scale one that we can run in the lab, run experiment on the lab and see how it performs in the lab. So I built this unit, a cyclone separator in the lab. It has about 2.8 liter of capacity. So I used 4 liter of leachate to run this experiment, and so it was flowing. It was a batch reaction. It was flowing, going through that, and leachate and solids were accumulating at the bottom. So it has these cone shape. If you see in the left picture, so this is the cone shape where this is the zone where the solids are accumulate, and we found out pretty good results. In just 10 minutes, we saw about 30% turbidity reduction as well as 70% TSS reduction in that experiment we have run. Now the issue is if you see the result at 15 minutes, the turbidity as well as the TSS increased again, which was the reason that the accumulation zone was too small for us, and we did not take out the accumulated solid after 10 minutes. So for this unit, about 2.8 liter with a 4 liter sample, it has about 10 minutes of time for solid accumulation without taking out the solid. So this is one of the things that we always have to take out the solids from the bottom of this so that more solids can accumulate without hampering my performance of the system. And we're planning to do that next. Now, based on this performance of this 30% of turbidity reduction as well as 70% of TSS reduction, if we consider as we know from the Solid Waste Authority data, about 6 million gallon per month of NEFCO flow, if we calculate that, how much solid monthly we have to handle, we wanted to calculate that based on the performance of our unit, small unit. Now we found out that it's about 100 pounds of solids per day based on the data, that experiment we have, about 100 pounds per day of solids we have to handle if we use the solid handling method. So yes, this method has definitely the potential of separating the solids from the NEFCO water, but anyway, we have to do some more analysis as well as maybe next time when we run the test, maybe build a new prototype with bigger solid accumulation zone. That could increase the performance too, as well as I can run the test for longer period of time, and that could help. And we're planning to do that in future. So another way, as I said earlier, the second way or second mechanism of forming this biological clogging or leachate clogging is the biologically induced clogging. And we wanted to check that in the field that if we remove the microbes from the leachate, what happens? So that was what we wanted to know. So this gives us this confidence in working that if there is no microbes and the precipitation still occurs, so there is microbes definitely may have some effect, but it's not the major impact that we wanted to know, that how it works. Now to do that, we plan to do UV disinfect leachate. We purchased with SW James. He helped us. He purchased the unit for us, and we tried this. We tried this into the field. So we have this field set up in the pump station A. If you have time, you can go visit that. So it is a 40 feet by 20 feet pipe network. We have used this during my master study, and for leachate clogging control with a flow what was the unit? Oh, sorry, the unit was an electronic scale control system. So we modified that unit, and we added this UV unit in the system and tried to disinfect the leachate and see how that impact the clogging. Now we have this on-site leachate reservoir, 900 gallons of leachate reservoir, and the flow I measured about 26 to 28 liter per minute. The flow was pretty good because our unit, it has 10 gallons per minute it can disinfect. The capacity of our UV unit was about 10 gallons per minute. So the flow is 26 to 28 liter per minute is about 9 gallons in that range. And our UV fluence was about 40 millijoule per centimeter square second, which is EPA standard for disinfecting, even drinking water. So it was pretty good fluence though. So we collected sample at different intervals and ran HPC count in the laboratory. So this was our one of the 3 experiments we have run. The result I have shown here, it's conducted on April 30, 2018. So the results there, you can see that they're very small, you cannot see, but those are not very important here. I'm not trying to focus on those. Those are just the data that shows that leachate quality has been consistent over the period I am running the experiment in both in treated or untreated site. But if you see the heterotophic plate count in the right bottom, shows that we did not really achieve much or significant amount of microbes or bacteria removal to say that the leachate was disinfected. And so that was one of the issue we were running through, and it could have been for several reasons that leachate has very low fluence, and it's very difficult for UV to pass through the leachate and disinfect. So that was one of the issues we found out after these experiments. But during this experiment, we have observed a very interesting question or question for our next research is that, so one of the days when I ran this experiment, and I came back after 7 days to check how much precipitation was formed after disinfection, I found out that the total system, the whole pipe network was submerged. Leachate was there. So I took out this 1 feet long, 1 inch dia bypass line where we were planning to measure the precipitation rate. I found that there is an orange reddish jelly-like substance in the surface of the pipe, and which was not there when I first tested and ran the experiment. So it was there, and I was like wondering what is this, and I took out these parts and measured the weight of these because I was trying to measure every 7 days the weight, how it's changing the weight of that 1 feet long pipe from the beginning to the end of my experiment. So next 7 days, after 7 days when I came back, the system was dry because I took out the pipe network, one feet long pipe. So there was no leachate anymore for the next 7 days. It was dry. And then when I came back, the same pipe, this jelly-like substance, this jelly-like layer, it forms a solid layer attached to the surface of that pipe, and it's not easily removable. I was trying to wash it, it was not going. It was attached to the surface, a layer of scale. Now that gave me this idea, this question. So it was jelly when it was submerged, and it is a solid layer when it is dry. Is this alternate submerged and dry condition is a big factor in leachate clogging, or forming in the leachate surface, or leachate precipitation forming in the layer of a pipe, inside of a pipe. So that is a big question. We will run a lot of experiment in the lab to answer, and that's very interesting for me to answer. So that's one very big thing we found out during this UV experiment, although UV didn't work, but it was a good observation to look at. So if it didn't work, it doesn't mean our experiment stops there. So we are planning to do still trying to understand how much impact has these microbes in the leachate precipitation or leachate clogging. So we have planned already, and we are doing some research on that too, that we will do heat sterilize the leachate so that we kill the bacteria, and then we address the difference in the sample before and the sample after, and then we run all the tests to check for precipitation potential and stuff. So yeah, UV didn't work, UV didn't disinfect our leachate, so now we will disinfect with heat sterilization and run all the tests we planned with UV. We will do that with heat sterilization so that we understand how much it impacts the leachate or leachate clogging. Now, pH has been always is a concern. Increasing pH definitely increases the leachate clogging. We have found out that everywhere. We know that, we have observed that, and literature also says the same thing. Now, pH in a landfill can change in different ways, and we are in our FAU lab, we are trying to look at into every aspects, every single way pH can change in a landfill leachate. And we want to understand how all these changes can change the precipitation potential over time, and how that impacts the precipitation rate. Now, pH can change with addition of carbon dioxide. We can reduce the pH. As I said, minus means reduction of pH, and plus means addition of pH here. Addition of acid, that can reduce the pH too. Sealing in open air, that increases the pH. We have observed that in our lab. Turbulence definitely increases the pH, addition definitely increases the pH, and addition of any base, that will also increases the pH. Now, we have this experimental setup in lab with carbon dioxide gas cylinders to see how, if we add carbon dioxide or added carbon dioxide into the leachate, how it changes the pH, how much it reduces the pH, and how that impacts the precipitation potential. So based on our experiment, we have found out that, so we have these gas, carbon dioxide gas, we put into a 2 liter of leachate at different rate, at 0.1 liter per minute to 1 liter per minute, and to see how that changes the pH of the leachate over time. And we have found out that in first 30 minutes, it reduces the pH by almost 2 to 3, 0.2 to 0.3 unit, and then the line becomes flat and it does not change anymore. So first 30 minutes of addition of carbon dioxide is definitely important, but it's after that, how much I add, it doesn't really change anything because it's already enough carbon dioxide in the system and it cannot take anymore. Now, with this experiment, we wanted to know that, so we reduces the pH of this leachate, and then we want to know how this reduction of pH impacts the precipitation afterwards, and we have that data after. We also looked into that as I have mentioned earlier, that leachate exposed to open air, it just exposed to open air, you don't have to do anything, just sitting open air, it's just increasing pH over time. So we also found out that it's about 30 minutes. After that, you keep it for 1 hour, it doesn't really change much. That first 30 minutes is important. So leachate sitting in a place for 30 minutes or even less than that is important rather than sitting in 2 hours. If it's 30 minutes already sit there, 2 hours doesn't really impact. It cannot sit there for 30 minutes. That is the issue. So that was one of the interesting findings also. Now, leachate exposed to mechanical turbulence also, there is, when leachate is flowing through the leachate collection system, there is definitely this turbulence, always there will be turbulence because there is a lot of, it's not exactly smooth. The leachate pipes is not exactly smooth, so there will be a lot of turbulence in there. Also, the leachate coming from different flows, we are adding dilution water in there, there is a lot of turbulence. How that impacts our pH of leachate that we wanted to know. So we see there a steady change over, I ran this test for 50 minutes, the data here for 50 minutes, and it was steady, it's increasing at 500 RPM. If we do some turbulence in the leachate, it increases the pH. And I ran this test, I remember, for 33 hours too, and it shows the increase, it's going up and up. So it's not that it'll go up always, but I think if I run it for 6 or 7 hours, there will be a point where it will be flat, and I think that pH would be around 8 or 8 point something, 8.2, where it will be flat. But the rate of change is slow, definitely, as you can see here from 0 to 50 minutes. It's about from 7.05 to 7.2. It's like 0.2 in 50 minutes. So if you consider that for 7 to 8, I have to run like at least 6-7 hours at 500 RPM, which will go at that 8 pH. After that, it can be flattened out. As I have said earlier, that all the leachate I have ran with carbon dioxide, and if you see the plot or the sample at the bottom, so this is leachate at pH reduced at different carbon dioxide rate. This is 0.1 liter per minute, this is 0.3 liter per minute, 0.5 liter per minute, and this is 1 liter per minute. And this was pH increased with aeration, just adding air to the pH, which increases the pH. To show the difference that if you have initial lower pH, that will have have lower precipitation rather than a leachate with higher pH at initial. And this is the control, so which we did not do anything with this. So we have total 6 samples. 1 is control, 1 pH increased with air, just addition of air, and 4 of the samples that pH we reduced with addition of carbon dioxide. During this experiment, so how we did this experiment, we have 200 ml of sample from all these conditions, control, addition of air, and addition of carbon dioxide, 200 ml sample each. We measured all the water quality parameters at day 0. And after that, we put this sample at incubator for 7 days at 35 degrees Celsius for 7 days, and take them out, measure how much precipitation was occurred in there, and also all the water quality parameter to check all the leachate precipitation potential too. So that was our total process of this experiment. Now, as you can see in the left top picture, so if you see that this is a layer of skin that floating in leachate that we found out, very interesting. Now, this I have never seen. I have seen that in the field, but I have never seen that in the lab, that a layer of leachate precipitation or cloud that was floating above the leachate. And and as you can see to the right, so this is the layer that was floating before. It's like a milk skin if you think of it, if you assume that, but it's in leachate, so it's different. And also there are some precipitation at the bottom. You can see the black ones. So there are two different type of precipitation occurring. One that is hard, that is attached to the surface, already attached to the surface with precipitation, and then there is this floating thing that can float anywhere and then can stack anywhere in the leachate collection system, and then can be a potential zone of this biorock or leachate clog formation. This can happen. Now, when we found these, and let's go through the water quality we have measured and how it's changed over the 7-day period. Now, pH, so initially, as you see, the control has around 7.1 pH, and we reduced with carbon dioxide all these, we reduced around 6.9 or 6.8 for sample, and one with air, we increased the pH. So initial pH, one sample with the air has about more than 8, around 8.2 pH, and our normal pH is around 7.1, so it increases, just addition of air increases the pH by 1 unit, almost 1 unit. So after 7 days, if you see all the pHs goes up, and it's between 7.54 and 7.85. That's what we found. The pHs increases over time if you just keep them at 35 degrees Celsius at 7 days. So that was pH changes over time. Now, alkalinity, initial alkalinity was around 2000. And as expected, it decreased over time. And as you can see, that alkalinity will decrease if the precipitation occurs much more. So for air, as you can see, the lowest alkalinity we observed for air, because the initial pH was higher, which results more precipitation. And that's why, so there will be less calcium and less carbonate in the leachate afterwards. So there will be less alkalinity in the system, so which makes sense. And also the calcium concentration, as I said, also it reduces from the initial concentration because of the precipitation, obviously. And we also measured the hydrogen sulfide here, which also reduced in 7 days. Now, we wanted to know the scale rate for all these 6 samples, how much scale is forming per liter of sample. Now, there are 2 types of scale we observed. Number 1, that is loose floating thin layer of scale I talked about earlier, and then the hard scale that is attached to the surface of the beaker. Now, we express this leachate scale rate formation in gram per liter, and also we have some questions here. So this is very interesting. Why this loose scale is forming, and then how does this loose scale is forming? Why this is floating? We don't know any answer of that, and I was looking into the literature, but I'm still struggling to find any reference that someone mentioned this loose scale floating, how this is forming, and why this is forming. What are the factors that affect in this loose forming and floating in the top, how this is floating. So that's very interesting to know, and would be one of my basic questions of my PhD to answer, I think. Now, how does it look? So this is the loose scale with 400x under microscope. It looks like that. It's not a mountain though, but it's the floating precipitate layer in our sample we found out there. And in the right, as you see the microbes, so that you can understand the difference, this is not a microbes. It's this thin layer of scale that formed. Now, we also ran some of the precipitation in the lab, controlled precipitation with higher pH. We adjusted the pH of the leachate with sodium hydroxide in the lab to precipitate the calcium carbonate and see how they looks. Now, these looks calcium carbonate precipitation in the lab with a different amount of sodium hydroxide. The goal of this was to understand that if that has something, this calcium carbonate precipitation has similarity with the scale formed earlier. Now, if you look in the edges of this structure, I know it's difficult to look at, but still we found some similarity between these formation and the edge of this precipitation. And also definitely we are running all these samples. We have all these samples collected. We are running through them XRD. So definitely we would know the concentration and as well as the composition of this sample later on. Now, as I mentioned earlier, the scale rate we found out definitely, as I mentioned, that air with higher initial pH will have more precipitation rate rather than a sample with control or a lower pH. Although the control theoretically control should have, as control had higher pH compared to all these three sample with lower pH, initial lower pH, but we need more tests, more experiments to refine these data. So this was a one sample, one time experiments. We have to run this 4 or 5 times to understand, to refine the scale rate. Although if I forgot to mention that this scale rate was calculated only on the scale that was attached to the surface, not considering the loose scale that was floating, because that was we have to find out a way or filtering it or any way to measure that how much is forming there. We did not consider that in our initial test, but we will definitely do in our next test. How much is loose scale, how much is the scale that is attached to the surface already in there, and that we will definitely identify in our next experiments. And one of our tasks was also the impact to the deep injection oil and how the scale and leachate clogging potential, how it's impacting the deep injection oil. So we always keep this mass balance analysis to keep us, as well as to share this data with Solid Waste Authority to give them the idea that the leachate water quality and how that precipitation potential, how it's changing over time. So we have this mass balance. As you can see, we estimate the mass balance based on all the leachate flow coming that we dump into the injection well, and I think it's difficult to see here. So it's about, we find out that in our calculation in the lab, based on all the water quality data we collect and the volume of each of the flow that goes through the deep injection well or weir well, we measure the Langley saturation index or the precipitation potential in other way. So precipitation potential in the model or the mass balance analysis we do in the field, we do in the lab, and then we also compare the data with the actual data collected in the field, and we compare them too. So how far we are, so to understand that. So yeah, we were pretty close here, about 1 in our calculation, 1.1 in the field. So we were pretty close that this mass balance definitely works, and we always keep this data up to date, and we will keep continuing doing it. Now, as Dr. Mirov was saying, we collected some samples from deep injection well with AECOM URS. We were being a part of that project too. So we found out that we collected 3 samples from 3 different depth at 0 to 200 feet, 200 to 1500 feet, and 1500 to 2900 feet. So we collected 3 samples there. We ran mineralogical analysis as well as some biological analysis ran by our biology department. And we found out that the water quality analysis shows that as we go down, the precipitation potential increases. We found out that, and increasing the LSI from 0.81 to 1.42. And we also found that the XRD analysis shows that we found out that solids we collected from those 3 depth, we found out quartz, silicon dioxide, calcite, and some salts and some magnetoferrite too. So finding salts, calcite, and quartz is pretty normal because there will be salt down there. And then there will be calcite definitely, because we have found out a lot of calcium carbonate going down there. It will be calcite, and quartz comes from sand mostly. So the results are pretty normal. And biological analysis, I have not done this. As I said, the biology department, the students involved and the professor involved there, they have done this. So we have found out that anaerobic mesophilic methanogens were the most prevalent species in there. And we also found sulfate-reducing bacteria, which is pretty normal. And we also found a biofilm generating non-pathogenic amoeba, which is a problem that we were trying to understand how to remove that so that it can not impact the flow going down the deep well. And also we found a virus there, which is Pseudomonas phage. We found that, and which kills the Pseudomonas bacteria down there. So in future work at Task 1, as I said, the impact of flow regime on geochemical rocking, we plan to run some experiment with varying Reynolds number based on different flow rate to see if that flow rate, how flow rate impacts the precipitation. as well as we will run more cyclone tests to separate solids and refine that data we produced before. And biological activity as clogging trigger mechanism, as we said earlier. So although the UVD didn't work, now we will move to the heat sterilization and run some experiment with that. And also that questions we want to answer, does alternate dry and submerged condition enhance clogging or scaling issues? So we will also try to answer that question. And as long as pH adjustment for enhancement precipitation control occurs, we are looking into all the possible ways that changes the pH of a leachate in a leachate collection system and try to understand the precipitation potential changes because of those changes. And impacts of deep injection well, we'll keep running all this mass balance we do all the time, keep updating the database, as well as we'll look into and as well as run some experiment in the lab to find out the pre-treatment of leachate as well as oil rehabilitation. And we always keep this task, and this is our favorite task, that perform other tasks for SW as requested by SW. Anything they ask, we are very happy to do that. So, and yeah, so key research find the key points here is if NEFCO can really be used as an alternative of groundwater, and if we can we use that? And the major issue remains that solid handling part. So we'll try to address that as much as we can. And then also, if rocking is biologically mediated, then we will definitely try to answer that by using that heat sterilization and see the impacts there. And most important questions for me and for my PhD is that how does this loose and hard scale is forming, and then to answer why this is floating or not sinking. So that's one of the questions I want to answer. And definitely as I said before, that oil rehabilitation as well as how to protect the deep injection well, that is one of our goal too. Thank you very much. If you have any questions, please. You're welcome. Sure. It's not starting. You can just come here.
Well, first of all, this is a very important study and a lot of good work, so it's exciting to see it. In your consideration about, I'm very interested in the biological aspect of leachate clogging. You said you're going to do the heat sterilization. I didn't know if you were going to also look at chemical treatment, whether it's acids or bases like chlorine. In groundwater wells, there's a lot of biofilming and scaling that goes on, and well drillers use chemical treatment as well. So, because it seems like pH is important, so if, let's say, you were to use an acid, which could also maybe kill the biological, you might also help prevent the clogging. So that's one question. And then the second question, in the lab, that black floaty, filmy stuff, were there microbes in there? I saw the microbe slide. Or was it all chemical precipitation? I'm curious to know if there's biofilming occurring. So, okay.
So for your first question, that if we are going to use any chemical for microbe or disinfection, so I think SW as well as many other facilities already use acids to reduce the pH or flushing of chemical clogging. And the issue with the acids is we can do that, and we have some analysis or some reports that came out maybe in 2016 that shows that we can reduce the pH of leachate by almost 1 unit by buffering capacity. We have some analysis on that buffer capacity. I think acids, I don't know exactly will it kill the bacteria, all of them acids, because I have to really go down, because pH, if I want to kill all these bacteria, maybe I have to go less than 4. Because they're 7, they're happy. At 6, they're not happy, it still works. At 5, they're still not happy, but still they will survive there, they will be there. Now the issue of acid is it's hazardous, and adding so much acids, it needs a lot of acids, and already the guys who operate there, it's a hazardous thing, And too much acids is always a problem. If you reduce the acids less than 3 or even 4 to kill all the bacteria, I think that would be an issue for all the operators, as well as when it goes down to the deep well. That for me, I don't think killing bacteria, acid would be a good option for killing bacteria. But I think chlorine, yes, we definitely can add chlorine to check how much it's disinfecting.
Well, is it working? Yeah.
Mhm.
So I don't know if Dan might be able to, Dan, do you do any well rehab work? Dan?
We do, and certainly chemical, adding of chemicals. Yeah, adding of chemicals in deep well treatment is common, certainly. I agree the acid problem becomes just a hazard to handle all that acid, and certainly expose it to the system is one that could cause more problems than good. The chlorine issue that you mentioned for bioscaling and other scaling and deep well injections, there are certainly that's been commonly used in the past, but again, other problems are created with carbonate formations that we're pumping all this into, and by introducing some of these chemicals. So I think there really needs to be a lot more research in this whole realm.
Well, and that's why I'm saying, you guys are doing research.
Mhm.
You could consider or take a look.
Sure, we will. Yes.
Yeah, I had one question just about the exposure to air in leachate systems, and obviously you've shown that this causes problems. What could we be doing? Do we need to be thinking at the design stage on how to keep air out of leachate collection systems more in general from the time, it's generated in the bottom of the landfill through its, extraction and placement into systems until it gets to its final injection location? What should we be doing to try to control that air exposure to air?
As we found out in these experiments that sitting in 30 minutes and sitting in a day doesn't make any difference. But we always want leachate to keep moving rather than sitting, not sitting for 5 minutes, not sitting for 1 minute. Keep it moving. As long as it's moving, that produces less problem than it's sitting. So we want it moving anyway, and as you have mentioned that if we have a way to not exposing all this leachate into air, that would definitely help to pH being in control rather than pH being going high. Anyway, so that would definitely help. And design stage, if we could do that, and that would definitely a point you should consider. And I think I did not answer your second question, which was that in my slide. So this one is so these microbes I just put in there to show that the formation of this floating layer was not related to microbes, rather than it's calcium carbonate precipitation. That's what I think from my experience, as well as all the XRD analysis I have run on the samples I have collected from SWA. Anyway, I will definitely run these through XRD to find out the exact composition of that.
It would be curious to look at the floating solids and the differences between the leachate.
Yes.
I think that's a volatile fatty acid issue,
Okay.
if I had to guess. But
Okay.
my big question was, you're talking about heat stabilization for
It's sterilization, yeah.
microbe removal. Calcium carbonate's inversely proportional to heat,
Heat.
so you're going to have an issue there.
Yeah, I know.
It will come out a solution.
So, yeah, thank you.
Go ahead.
Okay, so I was thinking. So when we talked about this heat sterilization, definitely we found that we increase the heat, calcium carbonate will be precipitated by that time when we do the sterilization already. But then we will have all these blanks and to address that problem. How much we are precipitating with the sterilization, and then the sample, and then we'll run the experiment with the blanks so that we understand how much this calcium carbonate is precipitated because of the heat sterilization, and then how much after that. So we'll address that problem issue.
And then my other question was, what's your theory on why the LSI is increasing down the well?
There was a difference.
Is
in,
there a temperature difference?
uh,
And it ties into
it's
my last
not
question.
not lot of temperature. I think it has different alkalinity and different amount of calcium and it's not only pH. So the pH difference, I think there was a difference in pH. I don't recall exactly what was the pH of the sample. One of the pH was a bit off, but other two were like pretty constant. So we expected all the three samples will have, I mean, equal or approximately equal pH, not changing too much. It's just all of them are under 200 feet or 1500 feet doesn't really change pH. We expected pH not to change, but the alkalinity was different, which definitely changes.
I'm
your
just
LSI.
curious if the temperature changed and you're seeing that change because of the temperature change.
We did not see a lot of temperature
Okay.
change. Yeah.
Hey, nice presentation.
Thank you.
With Dyer, I don't know if you've looked a lot at Dyer. Yeah, with the leachate, we average about 2 million gallons, a month. So over the course of the year, maybe 24, 25 million. Never had a rocking problem there. Class 3 is pretty clean. So it's limited to the Class 1. Do you think there's any merit to taking that leachate, Dyer leachate, and using that as part of the dilution on our Class 1 landfill?
I think that's always beneficial because dial leachate is, I think, I would say, has less concentration of everything compared to the class 1 that we collect leachate from. So that is not really creating a problem. That's really helping. More flow, more flushing. You're not using your groundwater. That's always beneficial. So just anyway, we are adding Dyer to the wet well. Why not adding that into the leachate collection system where it helps to push through all the leachate? So I think that's beneficial.
Yeah, just reroute it. Right now, I guess it's going on the backside.
Yeah,
Maybe
definitely.
reroute
We'll run
it
some
out
mass
to the Class
balance as
1
well
side.
as some analysis in the lab too to give you some good results on that too.
Yep.
But I think we have done Dyer mass balance in our previous studies. I think you have the report already. We have done that, but we can give you some updated results too on that.
Yep, very good.
I just want to share a couple of comments from Dr. Tiwari. He would like us to take a look at comparing the leachate deep well from the Solid Waste Authority, for example, with the one from Miami-Dade County at the Blackwater Treatment Plant.
Okay.
And then he wants to congratulate all of the young, energetic research scholars and their mentors for today's wonderful presentations. So with that, I think that concludes our presentations. And if there's any other questions for anybody, I'm happy to thank all the funding partners that we have for all of these projects, especially the Hinckley Center, and John Sherritt and Wes. And of course, the Solid Waste Authority, which is probably one of the best partners you can hope to ever imagine to have. They have such great expertise, and they're always so helpful in helping the students to graduate on time. And with that, I think their lunch will be served. So if you're online, go get lunch because we can't email you a sandwich. But for the rest of us here, we are ready to go. Thank you, everybody.
Thank you.